The main objective of Work Package 3 is the development and implementation of in vitro assays to test mixtures of concern on different immune cell populations. Together with our partners from Utrecht and Berlin, we aim to develop test strategies, so-called new approach methods or short NAMs, to test which immune cell populations are the most affected and what are the mechanisms behind the potential harmful effects of the chemicals that we test. In Work Package 3, we are actually focusing on both arms of the immune system. So we are focusing on innate immune cells such as dendritic cells, monocytes, natural killer cells, but of course we also focus on cells of the adaptive immune cells such as the B and the T cells. In Work Package 3, we initially planned to work first on a mixture of all immune cells together in blood derived from human beings and called PBMCs, and we will expose these PBMCs to our single chemicals or the chemicals and mixtures and study which immune cell population is the most sensitive within this mixture of all immune cells together. To investigate changes derived from the chemicals on the phenotype and the functionality of the single immune cell populations, we will use either well-established immune cell lines or primary immune cells. We will start with a mixture of all immune cells together derived from peripheral blood of human blood donors, the so-called PBMCs. We will expose these PBMCs either to our single chemicals or to chemical mixtures to define the immune cell population that is the most affected within the whole immune cell mixture. In our assays, we will either use well-established immune cell lines or primary immune cells. The primary immune cells will be obtained from PBMCs derived from human blood. We will isolate individual immune cell populations by magnetic activated cell sorting. Then these cells will be exposed to our either single chemicals or the chemical mixtures, and then we will study different endpoints. These endpoints will include general cellular parameters such as cell toxicity and proliferation, and also the capability of these cells to secrete more or less cytokines. Finally, we are also interested to study chemical-driven changes on the molecule level, so changes in expression patterns, and this will be addressed by transcriptomics and proteomics. Work Package 3 is mainly embedded between Work Package 2 and 4, however it's also indirectly connected to all the other work packages. We will receive the information from Work Package 2 of the ENDOMIX mixture to be analyzed in our assays, and then we will provide Work Package 4 with the information of the findings of our in vitro assays and also with the immune cells for their barrier models. Studies of single EDCs or EDC mixtures on the immune systems are still very underestimated, although immune cells are actually involved in all physiological processes of the body and therefore contribute significantly to the health status of the individual. We hope that our results will provide clear indications as to whether and to which extent EDCs are involved in the development of diseases. I personally believe that clear research findings are the basis for good political decisions and ultimately for the improvement of human health.